Hey, welcome back, everyone. We are going to finish up our discussion on meiosis and sexual reproduction by talking about specifically how meiosis is actually applied in a number of different organisms in the case of how they sexually reproduce. Now, this isn't going to be a very long or very extensive video. We're certainly not going to turn this into a sex ed lecture or anything like that. But I do want you to be aware that, especially based on what we've talked about and how we tend to apply a lot of these biological concepts specifically to humans, because that's just human nature, we care the most about ourselves. I want us to be aware that what we've talked about in humans does not necessarily apply to how other organisms go about their business. So let's go ahead and define something new here. A life cycle is defined as all the reproductive events that occur from one generation to the next similar generation. Now, again, if you're looking at this from a human point of view, that may seem like just about the most obvious thing in the world, but especially when we look at plants, you're going to see that a life cycle may include more than one generation. So we'll talk about that. So the only kind of life cycle we have discussed so far is the kind that we as humans and other animals go through. So in these cases, the organism is diploid, again meaning that all the somatic cells have two copies of each chromosome, and then the gametes are haploid. So a fertilization event in which we have a union between the two haploid gametes would then give you a diploid organism, which will grow and develop via mitosis. So we use meiosis for making the gametes, and we use mitosis for propagating the zygote into a fully fledged, fully formed multicellular organism. So that's how animals do it, but again, that's not necessarily how all organisms are going to do it. So let's consider how a lot of plants go about their business. We see what is called alternation of generations. Now, this is going to seem extremely unusual to the uninitiated. So, generations of plant alternate between a sporophyte diploid individual, that doesn't seem so unusual, and a gametophyte haploid individual. So, in the case of the gametophytes, the individual, the whole multicellular plant, is haploid in nature, meaning all the cells contain just one copy of each chromosome. So the idea here is, and we will see a picture to better illustrate this here in just a minute. The idea is that one generation of plant is going to be diploid, so it'll have two copies of each chromosome, and then that plant's offspring is only going to have one copy, and then the next generation after that is going to go back to being diploid. Now, one of your initial thoughts might be that, okay, well, the gametophytes are probably a lot smaller than the sporophytes. So you might be thinking that there's some correlation between how many chromosomes a cell has and how big the organism is or how complex the organism is. So back in the first video, we kind of already eliminated that possibility because we saw that there are organisms that have a lot more chromosomes than we as humans do. And we kind of tend to accept that we as humans are just about the most complex organisms that exist, even though we only have a total of 46 chromosomes. Now, we saw that a fern, again, which is a plant, has something north of 1,300 chromosomes, but we certainly don't go about thinking that plants or ferns are more complex than humans. So the same idea applies here. There's not necessarily a correlation between the size or complexity of an organism and how many chromosomes that organism has in each cell. And then another type of life cycle that we will see in a lot of fungi, it's basically the opposite of what we see in animals. The individual is haploid in nature, and the gametes end up being diploid. Now, if that seems very unusual, that's because it is, again, if the only thing you're used to is what we talked about before. So the individual is haploid. The individual organism has one copy of each chromosome in its cells, and then through gametogenesis, the formation of our gametes, those gametes become diploid, and then what you see from there is that the gametes undergo meiosis to get back to being haploid, and then they grow into an organism via mitosis. So for both alternation of generation in plants and then what we see here in fungi, we will see some illustrations that'll help us better understand it here in just a minute. Okay, so let's first start by talking about kind of the more conventional animal life cycle, which we are more familiar with at this point. So again, we are diploid organisms and we generate haploid gametes via a process called gametogenesis. Gametogenesis is not only going to involve meiosis of a type of diploid cell called a germ cell. So a germ cell, which is called a spermatogonium in males and then an oogonium in females. This diploid germ cell, which again has 2 copies of each chromosome, it will go through both meiosis 1 and meiosis 2 to produce kind of immature haploid gametes that then need to develop some of the physical characteristics necessary for fertilization. For example, a sperm, this spermatocyte that you can see right here, initially does not have its little flagellum that allows it to swim around and find an egg to fertilize. So after meiosis is complete, that development of that flagellum still needs to happen. So in both cases, both with spermatogenesis in men, which happens in the testes, and oogenesis in women, which happens in the ovarian cortex, we undergo meiosis of these diploid germ cells to produce our haploid gametes, which then through sexual intercourse allows those 2 gametes to interact with each other, and that's how you get your fertilization. The union of the chromosomes that are donated from the sperm to the egg that already has the 23 chromosomes from the mother. This produces a diploid zygote, which has a total of 46 chromosomes, and then through many rounds of mitosis and development and specialization and differentiation, produces a multicellular organism that will eventually grow and mature into organisms that can then do this whole process again. Now, something we're going to see on the next slide is that the manner in which sperm and egg actually go through meiosis is just a little bit different. So we've seen before that for one round of meiosis, which includes both nuclear divisions, if you start with 1 diploid germ cell, you're going to end up with 4 haploid zygotes, and that is, or 4 haploid gametes, and that is the case with spermatogenesis, but that is not the case for oogenesis. You are actually only going to produce 1 viable haploid gamete, 1 haploid egg. So we will see what that is all about, but we're going to see a new concept called asymmetric cell division in action here. So in the left diagram, you can see spermatogenesis, and there's really nothing too terribly interesting here outside of what we've already talked about. You can see your spermatogonium here. That is your diploid germ cell. It is going to go through meiosis 1 to produce our secondary spermatocytes here. And then it is going to go through another round of meiosis. That's the second nuclear division here, meiosis 2, to produce our haploid spermatids, which, like I said, then need to go through a maturation process to develop into mature sperm that are capable of participating in fertilization. So nothing too terribly interesting or out of the ordinary there based on our previous conversations. But now let's look at oogenesis. Here you can see the diploid germ cell, the oogonium. Now what is interesting here is that after meiosis 1, we are not going to form 2 daughter cells. We actually only have 1 daughter cell that actually retains the vast majority of the resources that the parent cell had. So the vast majority of the cytoplasm and membranes and organelles and proteins and nutrients. The 46 chromosomes that would have been segregated to the other daughter cell is contained within this structure here called a polar body, which is going to be extruded, and then it will wither off and disintegrate, and it will no longer be available to do anything. Meiosis 2, you're going to see very much the same thing. The vast majority of the resources are going to be retained in 1 cell, whereas we form a second polar body, which is then going to wither away and fade into nothingness as well. So this is a case, like I said, of what you call asymmetric cell division, in which instead of symmetric cell division, where we try to divvy up the materials equally between the 2 daughter cells, In this case, with asymmetric cell division, we instead keep the majority of the resources in one cell, leaving the other cells to not have enough resources to survive, which is why those polar bodies eventually wither away and die. Now, another thing that is very interesting about oogenesis, but we're not going to get too deep into it because it's probably a topic better served for human physiology, and I do have a video on my YouTube channel talking about this very thing. You'll want to go searching for my Human Physiology Chapter 27 videos for more information on both of these processes. But something that's very interesting about oogenesis is this is not a single continuous process. The first part of this, up from the division of the oogonium into a primary oocyte, and then the beginning of the entry into meiosis 1, this actually happens before a woman is ever born. So when a female is conceived and then is developing inside her mother's uterus, this first step in the process is already occurring for that fetus. So once you get to prophase 1 of meiosis 1, the whole process gets paused and does not resume until that little girl is born and until she grows up and hits puberty. Then the process resumes, and then we're going to hit another pause button halfway through meiosis 2, and that process will not resume until a sperm actually comes in and fertilizes the egg, and then the remainder of meiosis will actually occur. So very interesting, not something you specifically need to know for your study of general biology, but if you're interested, you can look more into that with those videos that I mentioned before. Okay, now here is the alternation of generation process that we often see in plant cells. So you can see that the entire life cycle includes two different generations. You have a generation that is diploid in nature, and we of course call these sporophytes. And in producing their gametes, they form what are called sporongia, which then develop into spores that then obviously are going to be haploid in nature. And then those spores are able to germinate and then divide via mitosis to produce a haploid individual, which again we call a gametophyte. Now, the gametophyte here is going to sexually reproduce in the way that we are a little bit more familiar with. The plant will have both male and female components to it, and you can see here the quote-unquote male gametes are going to come around and fertilize the female component, the egg here. So very much like what we see in animals, we get a fertilization event, which is going to produce a diploid zygote, which through mitosis will get us back to a sporophyte and diploid individual. So the very interesting thing about this alternation of generations is that the manner in which we make our gametes and then develop them into an organism is totally different depending on which generation you start in and which generation you end up in. And then here you can see what goes on in a lot of fungi. You can see that the individual, as you see it down here, is haploid in nature. But if we kind of follow the process around, and you can see there's kind of some complicating issues here. But if you kind of pay attention to the most important components of this, once we form our gametes, the gametes eventually are going to end up as diploid in nature, which we call zygospores. And then they are going to undergo meiosis to form haploid spores, which then will divide via mitosis to produce a multicellular fungal species. All right, so that is going to do it for this little survey into different types of life cycles and different manners in which different organisms undergo sexual reproduction. We didn't really get into the weeds on any particular topic, so if you find any of these topics particularly interesting, I invite you to look into them a little bit more on your own. But the main thing to take away from this is that not every organism is going to reproduce in the same way, and we've certainly already seen this in the case of prokaryotes, which undergo asexual reproduction, a manner in which you should end up with daughter cells that are genetically identical, whether that be through mitosis or through binary fission for prokaryotes. But we've said before that for sexual reproduction and any process that involves meiosis, generating genetic diversity is extremely important. All right, that is going to wrap it up for this chapter. In the next few chapters, 12, 13, and 14, we are going to start looking at DNA structure and function, and we are going to start looking at chromosomal patterns of inheritance, which is going to cause us to dive deep into the experiments of Gregor Mendel, which you could argue are some of the most important experiments and most revealing experiments in the whole history of biology. So I hope you'll be looking forward to that, and I hope you'll join me then. Thanks a lot, and see you later.